My name is Floris van Delft. I am a part-time professor in bioconjugate chemistry at the Wageningen University. Besides, my main affiliation lies with Synaffix, a small biotech company located in Os. Synaffix offers the technology for the generation of a specific class of anti-cancer drugs, so-called antibody drug conjugates, which is the topic of this lecture. As you will learn, the topic of today fits seamlessly into the course Applied Biocatalysis. However, not so much to generate bulk materials or products in a cheaper or more environmentally friendly way, but today you will learn how enzymes can be employed to generate real high-value goods, like medicinal drugs, for the targeted cancer therapy. So the topics of today are an introduction to antibody-drug conjugates, some general perspectives on enzymes and how they can be employed for specific protein functionalization. One topic that deserves special attention is click chemistry, and finally I will explain to you how you should approach the case studies for this topic. Okay, so let's begin with an introduction to antibody-drug conjugates. So antibody-drug conjugates can be traced back to the late 19th century when Paul Ehrlich in Germany formulated the concept of the magic bullet theory. Basically, he was a visionary man because he envisaged how pathogens would be attacking cells of a host organism, in this case a human being, by binding to receptors located at the cell periphery. And the cell would be responding to this by shedding excess amount of the cell surface receptor, so the pathogen would be bound in circulation and would no longer be able to reach the cell anymore. So building on this concept, you can envisage how you can have an antibody type molecule that would be binding to a pathogen and bind to it or kill it before it actually reaches the host cell. Building on this concept of the magic bullet theory, we here see an artist impression of a cell surface, which you see is really highly decorated with a large number of biomolecules. These can be glycans, these can be proteins, these can be lipids, and all of these receptor molecules are present in a certain quantity. And the quantity of these receptors can be different on a tumor cell versus a healthy cell. And if you take that as a basic consideration, you can imagine how it would be possible to make use of the exquisite selectivity of antibodies depicted here, which are able to bind with high affinity and high selectivity to a tumor cell, but not to healthy cells. Then, if you charge this antibody with a very toxic molecule attached to the antibody via a covalent linker, you can imagine that this antibody could drag the toxin to the tumor cell and then in a Trojan horse-like mechanism be taken up by the tumor cell, taking into the cell the toxin and thereby leading to apoptosis of the tumor cell. In this animation, you see this Trojan horse-like mechanism depicted in a clear fashion. So first, the antibody-drug conjugate is binding to a specific cell surface receptor on the tumor cell. Next, this receptor is taken up by the tumor cell via endocytosis, taking along the ADC. Once into the lysosome, the ADC will be processed, breaking down, thereby leading to liberation of the cytotoxic agent, which is released in the cytoplasm. Then the cytotoxic agent will induce apoptosis of the tumor cell. Now I can imagine you have never heard of antibody-drug conjugates or ADCs, but these are really a rapidly emerging class of therapeutic drugs for targeted treatment of cancer. For example, look at the number of ADCs that have been approved in recent years, starting in 2011 with Adcetris, in 2013 with Kadcyla. After that it was silent for a few years, but since 2017 an additional 7 ADCs have been approved, and there are many more, approximately 80, currently under clinical evaluation. So you see the number of ADCs is rapidly increasing, and they are establishing themselves very strongly in the field of anti-cancer therapy. Now, if we look a little bit more closely how these ADCs are prepared, it becomes clear that there are basically two synthetic approaches for that. Either by attachment of the cytotoxic molecule to the native lysine side chain or to native cysteine side chain. As you will see in the next slide, both of these approaches really lead to mixtures of products that do not have the superior properties that one could expect, so there's really room for improvement. And this becomes clear that if we do an analysis of the pipeline of the ADCs that are currently under clinical development, we can identify a number of approaches that are leading to more stable and more homogeneous ADCs, of which enzymatic approaches are really the fastest emerging, with currently 7 ADCs under clinical development. That's why this is the topic of this course, and that's why this will be the topic of the case studies that you will perform. So let us look at Adcetris, the first approved ADC. And Adcetris was approved for treatment of non-Hodgkin lymphoma and anaplastic large cell lymphoma, and it consists, as all ADCs, of an antibody conjugated to a cytotoxic molecule. Here you see the chemical structure of Adcetris. You see it's an ADC prepared by conjugation of a cytotoxic molecule to the native cysteine of the antibody. The antibody, in this case, is binding to a target, CD30, which is highly expressed on B cells that is leading to these diseases. Attached to the cysteine is a cleavable molecule. You see here that dipeptide, which is actually cleaved once it's inside the tumor lysosome. Then it releases the cytotoxic molecule called MMAE, which is a tubulin binder, which eventually leads to apoptosis of the cell. Now, if we zoom in on the analytics of the ADC, we see very clearly that it's the mixture of components, which is not surprising if you consider that there are 8 cysteine side chains that are conjugated, but on average only 4 of those are conjugated. So it's really a stochastic distribution of a mixture of components, and you see the HPLC profile depicted here at the bottom. Now, is that a problem? Yes, it is, because some of these antibodies will not be conjugated at all and only lead to blocking of the receptor, but preventing conjugated antibody ADCs of binding and internalization. Also, part of the component is really highly conjugated, very hydrophobic molecule, which will be rapidly cleared from circulation, and thereby also compromise the activity of the drug. Okay, on to our next topic, how enzymes can be employed for antibody modification. So I guess it's fair to say that in the past enzymes have mostly been used for analytical purposes. As you may know, an antibody is a 150 kilodalton protein existing of a heavy chain and a light chain, many amino acids. It's also glycosylated, and enzymes have been very useful for the analysis of antibodies. For example, one can take papain, which may or may not be immobilized, and the papain will digest an antibody and cleave the heavy chain just above the disulfide hinge region, thereby cutting off the Fab fragment of the Fc fragment of the antibody. The Fc fragment can be bound to protein A resin, and then the Fab fragment can be analyzed. A very similar enzyme is called IDS, and IDS will actually trim just below the hinge region of the antibody. So here the Fab2 fragment is liberated and the 2 separate Fc fragments. Actually, I forgot to mention trypsin. Trypsin may be familiar to you because trypsin will digest any protein and cleave the covalent bond after any lysine or arginine amino acid, thereby providing a large number of protein fragments for mass spec analysis. A fourth enzyme that is worth mentioning is PNGSF, because PNGSF will actually trim any N-glycan of any protein. Because all monoclonal antibodies are glycosylated, the glycan will be liberated and the glycan can be analyzed then separately. Now, I just mentioned that enzymes are mostly used for analytical application. However, there is in fact one marketed drug that is prepared by papain digestion of an IgG1. And this drug is called RioPro. That's the trade name, or the generic name is apiximab. So this is the first FDA-approved antibody therapeutic. It was approved in 1994 and was developed by Johnson & Johnson and is now marketed by Eli Lilly. So RioPro is binding to glycoprotein 2B, 3A, that is present on platelet membranes. And in doing so, it inhibits platelet aggregation that is often taking place after coronary artery procedures. So you see, this is a relevant example of the use of enzymes for development of medicinal drugs. Now, if we look at the enzymes used for commercial protein modification, this can actually be categorized into two sections: non-pharmaceutical applications and primarily pharmaceutical applications. The non-pharmaceutical applications are characterized by protein cross-linking enzymes. For example, microbial-transglutaminase, lacase, peroxidase, or other oxidizing enzymes will lead to protein polymers, which can be useful in the food industry, leather industry, textile industry. If we look at the pharmaceutical application, the enzymes can be used for attachments of a molecule to a protein, and this can be a small molecule, this can be a carbohydrate, or this can be a polymer. You see here a long list of enzymes that can be applied for that I will not go through now, but you can look at them yourself. And some of these are actually applied in ADC context. So the concept explained in the previous slide, you see this here graphically depicted. For example, proteins can be dimerized or polymerized. A synthetic polymer can be attached to a protein, which will be explained in the forthcoming slides. Protein glycosylation can be performed selectively, as we saw in the example of apiximab. A small molecule can be ligated to a protein, for example, used in ADC context, and also small polypeptides or DNA fragments can be attached to proteins of interest. Next topic, protein functionalization. You just learned how enzymes can be used for protein modification. However, besides enzymatic methods, we also know a vast array of chemical procedures for protein modifications that are basically based on the modification of the native amino acid side chain. Most prominently, these are the lysine side chain and the cysteine side chain, because the cysteine is the only amino acid that has a free diol, and lysine is the only amino acid that has a free amino group, both of which are functionalities that rapidly undergo chemical conjugation processes, like alkylation or disulfide formation for a free diol, or acylation or alkylation or reductive amination for the lysine side chain. Besides, in some cases, the N-terminus of an amino acid of a protein can also be used for controlled modification, and there are examples for tyrosine modification. One chemical method that is often applied for protein modification is so-called pegylation. Pegylation stands for the attachment of a polyethylene glycol chain to a protein. And polyethylene glycol, you see the structure of which is depicted here on top. So it's a polymer of ethylene glycol, which may exist of 20 or 50 or even more units. By attachment of a polyethylene glycol chain to a protein, several advantages occur. For example, solubility can increase, but also the half-life of the protein, if in circulation, if used for pharmaceutical application, can significantly increase because the uptake by the kidney is reduced. Thirdly, proteolytic degradation of the protein can be reduced by attachment of a polyethylene glycol chain. So pegylation of proteins really isn't an anomaly. In fact, it's an enormous market that is comprised by a multitude of pegylated proteins you see depicted here in this table. The yearly revenue of some of these pegylated proteins can exceed more than $1 billion, so these are really blockbuster drugs. One notable example of a pegylated protein is Longprex, with the generic name lipexfilgrastim. This drug was approved in 2013, brought into the market by Teva, and is acting as a long-acting version of the human granulocyte colony stimulating factor. This is a glycoprotein that stimulates the bone marrow to produce granulocytes and stem cells and releases them into the bloodstream. So by pegylation of the protein, the circulation time of this GCSF is significantly enhanced and thereby applied for the sustained treatment of chemotherapy-induced neutropenia. At the bottom you see how this pegylated protein is in fact produced. So first there's a recombinant expression of the protein, which is O-glycosylated by serine or threonine. This O-glycosylated protein is subjected to a mixture of a GalNAc transferase and UDP GalNAc. So thereby the GalNAc is attached to the terminal serine or threonine. Next, the GalNAcylated protein is again subjected to a mixture, in this case by a sialyl transferase, in the presence of a pegylated sialic acid. So effectively hereby the protein is pegylated in a really controlled fashion. The final topic of this course is click chemistry. This is a bit remote from the topics that were discussed before, but the relevance of it will become clear to some of you during the preparation of your case studies. Click chemistry was coined as a term by Nobel Prize laureate Barry Sharpless in 2001. It basically denotes a subclass of all chemical reactions, but only those that qualify and fulfill a set of very stringent conditions. For example, the reaction has to be extremely selective. It has to be able to operate at neutral pH at room temperature. It has to be fast and irreversible. And maybe most difficultly, it also has to be performed in water. Click chemistry can be thought of as a reaction between two molecules that have no choice but to react with each other. This is an analogy with the movie Fatal Attraction with Glenn Close and Michael Douglas, where you can imagine that happened in this movie. So if we think of Glenn Close as a cyclooctane molecule, and if we think of Michael Douglas as an azide, these two molecules will react with each other and form a very strong bond very quickly. So why is the reaction of a cyclooctane and an azide so extremely fast? Well, maybe you remember from your first year organic chemistry course that a carbon in a triple bond is sp hybridized. And sp hybridization dictates that the bond angle of this carbon between the triple bond and the single bond is really 180 degrees. So you can imagine what happens if you bend the molecule so the bond cannot be 180 degrees, but in fact, like in an 8-membered ring, is rather like 157 degrees. This is a molecule with a very strong strain on the triple bond. So this triple bond is very likely to react. And if this triple bond encounters an azide, and you see the structure depicted here, then quickly a so-called triazole will be formed, which is an irreversible reaction. So click chemistry is really a bucket name for a family of reactions that are really characterized by the presence of a strained system in the reaction. Typically this will be a cycloalkyne or a trans-cycloalkene, both of which have very high strain. So the slower of these reactions are really the reaction of cycloalkynes with azides. However, if a cycloalkyne is reacted with a kenone, or a trans-cyclooctene is reacted with a tetrazine, the reaction rate goes up a factor 100 or 1,000, or even it can even go up with a factor 1,000,000. Finally, let's take a minute how the case studies should be approached. So this slide highlights ADCT 601. ADCT 601 is an ADC under development by a company called ADC Therapeutics, which has taken a license from Synaffix. ADCT 601 is an ADC that is currently in phase 1 clinical studies, and it's an ADC that targets AXL1. It's a receptor that's highly overexpressed on solid tumors. To the antibody is attached an extremely cytotoxic payload called perilobenzodiazepine, also known as PBD dimer. So how is ADCT 601 made? So this is a combination of enzymes and click chemistry. In phase 1, a set of 2 enzymes will remodel the native glycan on an antibody, thereby trimming the most part of the glycan and reintroducing an in situ modified sugar. Then in the second stage, the cytotoxic payload is attached using this metal-free click chemistry, the reaction between the cyclooctane and the azide that has been installed on the enzyme, on the product, on the antibody. So in this overall 2-stage process, the antibody is modified with exactly 2 molecules of the PBD dimer. As said, ADCT-601 serves as an example how enzyme and click chemistry can be used to make antibody-drug conjugates, or more general, how an enzyme can be used for the controlled modification of an antibody. So the assignment for your case study will be, with your group, to look into 3 different aspects of enzyme modification of antibodies. First, you will look into the background of a specific enzyme. Second, you will research the commercial application of this enzyme for a particular application. And thirdly, you will discuss in detail a case study of an antibody-drug conjugate that is currently in the clinic. I will be available for supervision and support. So you find my email here on this slide. Also, during the afternoons, you can call me on my cell phone, and I'm happy to assist you if required. I wish you good luck, and I look forward to your case studies.